Microtubule networks must be precisely arranged within cells for them to carry out their many functions. One way that cells organize the cytoskeleton is by anchoring the plus ends of microtubules to the cell cortex, a feat accomplished by plus end binding proteins like the clasps. Clasp proteins attach to the cortex via a pH domain containing protein called LL5-beta. In the May 31st issue of the JCB, a team of scientists led by Yuko Mimori-Kiyosue from the Riken Center for Developmental Biology in Kobe, Japan, examine how LL5-beta and its close relative LL5-alpha specifically organize microtubules at the basal surface of polarized epithelial cells.
We found that LL5-beta as well as LL5-alpha are localized at the basal cortex of epithelial cells. For a long time, it has been known that microtubules in epithelial cells are aligned along the apical-basal axis with first ends facing towards the basal side. However, the molecular mechanisms underlying the organization of these epithelial microtubule networks have remained to be clarified. We decided to analyze the function of LL5s in organizing epithelial microtubules.
Mimori-Kiyosue and colleagues found that clasp-bound microtubules failed to attach to the basal cortex of breast epithelial cells when LL5-alpha and beta were both depleted. But how are the LL5s themselves recruited to the base of cells? Live cell imaging revealed that LL5s accumulate basally as epithelial cells attach to their underlying substratum, suggesting that LL5s might localize to integrin-based cell adhesions.
LL5s never co-localized with focal adhesions. However, interference reflection microscopy, which visualizes cell-to-substratum attachment, revealed that LL5s were distributed at cell adhesions, shown as gray zones, and LL5s co-localized with laminin depositions.
Laminin is secreted by cells, which then bind to the extracellular matrix protein through the integrins alpha-3-beta-1 and alpha-6-beta-4. LL5s, laminin, and these two integrins all co-localized in epithelial cells, and at the basal surface of mouse epithelial tissues.
We hypothesized that cell-to-substratum attachment may trigger the accumulation of LL5 at the basal cortex.
To see whether laminin and the laminin receptor integrins do control LL5's recruitment to the basal cortex, the researchers grew epithelial cells in 3D culture. The cells form balls in which LL5s, laminin, and the integrins all co-localized on the outer basal surface.
We knocked down integrin subunit separately or in combination. When integrin alpha-3, alpha-6, beta-1, or beta-4 was separately knocked down, we observed only slight decrease in LL5 accumulation at the basal cortex. However, when integrin alpha-3 and alpha-6 or beta-1 and beta-4 was simultaneously depleted, LL5s dramatically reduced at the basal cortex.
LL5s therefore depend on the laminin receptor integrins for their basal localization. Indeed, their accumulation is initiated by integrin signaling. An antibody that artificially activates integrin alpha-3 induced LL5's recruitment, as did laminin-coated microbeads. But LL5s also affect integrin distribution. Knocking down LL5s reduced integrin alpha-3's localization to the basal cortex of epithelial cells, although integrin alpha-6 was unaffected.
We then expected that LL5 form a complex with integrins, so performed pull-down assays of LL5s and the cytoplasmic tails of integrins in cells, and indeed we observed their associations. However, we observed no positive result in Beast2 hybrid assays and in vitro GSD pull-down assays, indicating that their interaction may be indirect.
So LL5s are recruited to laminin integrin-based adhesions in epithelial cells. What happens then to the cell's microtubules when LL5's basal recruitment is disrupted?
We set up a system to visualize microtubule dynamics in 3 dimensions using EB1GFP, a marker for growing microtubule end. Analysis of the three-dimensional distribution of EB1-GFP revealed that microtubule growth frequently occurred near the apical and basal field surfaces. After LL5s were knocked down, the number of growing microtubules along the basal cortex was selectively reduced, whereas it was increased in other cell regions. Furthermore, the intensity of microtubule staining was reduced at the basal cortex and increased in the upper part of the cells.
Knocking down laminin or its receptor integrins had the same effect. Microtubules were selectively reduced in the basal region of cells. The recruitment of LL5 to laminin integrin adhesions is therefore required to maintain a dense array of microtubules targeting the basal side of epithelial cells. Other microtubule stabilizing proteins such as ACF7 and APC remain at the basal cortex in the absence of integrins alpha-3 beta-1 or alpha-6 beta-4, suggesting that there are several pathways that anchor microtubules at the basal cell surface, allowing epithelial cells to align their microtubules along their apical-basal axes. So why do polarized epithelial cells organize their microtubules in this way?
This is still an open question. Microtubule alignment could be beneficial for the directional transport of specific cell components by plus-end directed motors. However, no specific cargoes transported exclusively towards the basal cortex have been reported. To clarify the importance of appropriate organization of microtubules in polarized cells, I am going to analyze the function of microtubule organizing factors during tissue morphogenesis and development.
In the meantime, you can read more about how signaling through laminin integrin adhesions recruits LL5s and microtubules to the basal cortex of epithelial cells in the paper by Hotta et al., published in the May 31st issue of the Journal of Cell Biology.